All right, Bio 35. So this is your immunity lecture. Let's see, new unit. Okay. All right. Blood typing. So all immunity, blood typing comes under the immunity topic. So, there's 4 types of blood, 4 blood types. Now, like this, the blood types are determined by the type of membrane proteins on the surface of your red blood cells. Right so far. And if you have the A-type antigen on the surface of your red blood cells, that's determined by a gene. You've inherited a gene that makes an A-type membrane protein, that's blood type A. If you have the B-type antigen on your red blood cell membrane surfaces, you have blood type B. That's another gene. If you have both genes, we inherit 2 genes for everything, one from our mother and one from our father for common traits, like hair color or eye color or height or personality or blood types. Say you get an A gene from your father and a B gene from your mother, you end up with A, B, type AB blood. And if you inherit the gene for neither A or B, then you have type O blood, and you're missing those antigens. Okay, now there's also another potential membrane protein that could be on your red blood cells, and that was labeled as the RH factor. RH stands for rhesus. The rhesus monkey, they studied the blood types. They figured out the blood types, the RH factor blood types in rhesus monkeys first. And so they devoted it to them. So any, type A blood could be RH positive. If you have the factor, if you have the membrane protein, you're considered RH positive. And if you don't have the factor, you're RH negative. I'm going to remember to unplug the phone this time. Okay, right back. It always seems to ring right in the middle of my lecture there. Okay. So we were saying if you have the RH factor, then you're RH positive. If you're missing the factor, you inherit the gene that doesn't make the protein, then you're RH negative. So any of these blood types could be Rh positive or Rh negative. So in reality, there's 8 blood types. A positive, A negative, B positive, B negative, AB positive, AB negative, and O positive and O negative. And the most common blood type is O positive, and the most uncommon blood type is AB negative. Most folks are Rh positive, like 85%, give or take, 15% being Rh negative. So, and O is the most common blood type, amazingly, even though it's a recessive gene, it's the most common blood type. So the positive is the most common type of Rh factor, and O is the most common type of blood, so O positive is most common. AB is the least common blood type, and then of course the negative factor is only 15%, so AB negative is the least common overall blood type. It seems simple so far, but then it gets complicated when you start to study the types of antibodies that are in the plasma for everybody's blood. And so everybody has the opposite antibody in their plasma, which makes sense. You don't want the antibody that's going to react with your own blood, then you'd be destroying your own blood cells. So in other words, the A-type blood people, they have the anti-B antibody, from birth on in their blood plasma. The B folks have anti-A antibodies in their plasma floating around. The AB folks, do they have those antibodies? They hopefully not, because if they did, then those antibodies would react there, so they have none. And the O blood has both anti-A and anti-B antibodies in the blood plasma. So when you're giving a transfusion to somebody, you have to take into account, the medical profession has to take into account not only the types of antigens on the red blood cell membranes, but you have to take into account the type of antibodies that are in the plasma. So what they've learned to do is when giving blood, they now kind of remove the plasma, which has the antibodies in it, and then keep the cells and just use the cells, and then give for the fluid. If you're giving somebody, you want to give them whole blood, well, rather than doing that, you give them the cells, wash away the plasma, remove the plasma, and then give them a normal saline IV solution to build the blood volume. So you give them the cells, we call those packed cells, and then an IV solution. Okay, it's a little bit, that's easier to match. So that's what we just did here. The antigens are those red blood cell membrane proteins. They represent those with an I. So you inherit 2 genes. Like I say, you inherit 2 genes for all traits, all characteristics. So we inherit, 1 gene from our mother and 1 gene from our father for our blood type. And that's called our genotype. If you were to study what kind of genes do you have for your red blood cells, that's a study of your genotype, your blood phenotype. The phenotype is the actual type of blood that you have, the actual physical type of blood, whether you have A blood, B blood, AB blood, or O blood. That's the phenotype, the physical type. So to be type A blood, type A, you could have inherited 2 A genes, or you could have inherited an A gene and an O gene. They represent that by a little I. They represent the O gene with a little I. Not sure why, but they do it with a little I, lowercase I, like that. The O gene is a recessive gene, always, to the A and the B genes. So if you have inherited the A gene or the B gene, then those antigens will be developed, and they will be presented on your red blood cell membranes. They will be present. The O gene will basically then be hidden. It's a recessive hidden gene. So the only way you can really be O is if you inherited both O genes, because they're both recessive. So you inherit an O gene from your mother and an O gene from your father and your type O blood. So you would think that type O blood would not be that common, but it is the most common, amazingly. Type B blood, then, those folks, then what could they be? They inherited both B genes, or they inherited the B gene and the recessive O gene, type B blood. That's their genotype, their potential genotypes. Type AB, they have to inherit this. They inherited the B gene from one of their parents and the A gene from the other parent, like that, AB. And then type O, I mentioned earlier that they must inherit the two O genes to be type O. The RH factor is much the same. The RH positive is the dominant gene and RH negative is the recessive gene. So that's why most people are 85% are RH positive. Okay. All right, let's see what we got in here. Here's about the antibodies I mentioned earlier. Universal recipient is AB positive, is considered AB positive. They have all of they have the A antigen, they have the B antigen, they have the RH factor antigen. So they have all the antigens. Their immune system recognizes all of those and doesn't attack them. You know, the immune system attacks foreign antigens, something that's foreign to your body. So if you had type A blood and you were given type B blood, that's a foreign antigen. And so your immune system would recognize that and attack it. So you have to match blood. So AB positive actually is the universal recipient. O negative would be the universal donor. When you donate that blood without the plasma, just the cells, without the plasma, you're giving them cells that don't have any antigens, so that the recipient's body doesn't reject it, and it doesn't have any of the RH factor antigens either. So you're not giving them any foreign antigen. So O negative is the universal donor. Now, so here's what I was saying. If you were type A blood, there's the A antigen on the surface, and you were given type B blood. Here's type B blood. And you were given type B blood. They have the antibody for type A antigens, right? If you were given whole blood. So that person's, the donor's plasma anti-B antibodies would then react with your type A blood cells, and it would cause them to stick together like that. They would agglutinate, so it would plug up all your blood vessels. So that's why you gotta match. Same thing with the other. If you had type B blood and you were given type A blood that had the plasma with the anti-A, anti-B antibodies, they would react with your type B blood and agglutinate it. Sorry, it's down here. It's got all these arrows here. Agglutinate it like that. So in other words, if you were type A blood and you were given type B blood, the antibodies in the type B blood plasma would react with your blood cells and agglutinate them, plug up your blood vessels. So you have to match. Here's what agglutination looks like. There's no agglutination. There is agglutination there. That's the antibodies clumping up the red blood cells, sticking them together. That's got agglutination. That's no agglutination. There's agglutination, and that's agglutination there. Whenever so this is blood that's been added anti-B or anti-A antibodies. Okay, so this is type A blood here. It's type A blood. You see the A antibody that we gave that person the A antibodies. They clumped up their blood, so that's showing that, oh, they do have type A blood. See, here's the clumping in the anti-B antibodies. not there, but here. So that's showing that they do have the type B antigens because they clumped with the antibodies. Here's the type AB blood where their blood agglutinated with both test serums. You know, the anti-B test serum and the anti-A test serum agglutinated both times there. So that's AB blood. If you had no agglutination with either test serum, what type blood would you have? You have O. You know, the Rh factor, they actually call it antigen D. Some of the Rh positive people have the antigen. Rh negative do not. The pregnancy thing is if you're a female with Rh negative blood and you have your male partner was Rh positive blood, the fetus could end up being Rh positive. And the mother is Rh negative. So the fetus is inside of you, Rh positive, and the mother is Rh negative. Here's the thing that could happen. It's erythroblastosis fetalis. It's this. If this is mom, here's mom, Rh negative. Here's baby, Rh positive. If somewhere along the pregnancy, the bloods were to mix, there was an injury or something where the maternal blood and the fetal blood actually mixed, then mom would have been exposed to this Rh positive antigen, which is foreign to her body. So her immune system would respond by making anti-Rh antibodies. During the pregnancy, anti-Rh antibodies. And so as those are being produced, they actually are small enough to cross the placental barrier and they attack the fetus's Rh positive blood. And so that, depending upon when that happens during pregnancy, if it was early on, it could be fatal to the fetus. It's easily dealt with though. If you're Rh negative, you're a female and you're Rh negative, they just give you a shot during pregnancy and it can prevent that whole process from happening. Very good. Okay, so we're going to do the rest of immunity here on the board. I'm just going to move you over here. Look at the board. Me and the board here. I'll move you over and give you the better view, best view possible. All right, so here we are on the board for the remainder here. I'll get you more light. Don't want you to be able to see me too much. All right, immunity, non-specific immunity. And specific immunity. Well, so non-specific is when, it's also called innate, by the way. Your author might call it innate immunity. That's immunity to many different things all at once, like your skin, say. Your skin is a barrier to lots of different types of pathogens. Okay, that would be an example of non-specific immunity, barriers. Specific immunity then is when your body makes an antibody specifically for a certain pathogen, or it makes a cytotoxic T cell designed to kill a specific pathogen, a certain pathogen, not a bunch of them, just this certain one. That's specific immunity. That's sometimes called adaptive immunity. This would be innate, and this would be adaptive immunity. Okay. A pathogen. I mentioned that word pathogen. What is a pathogen? You guys name what the pathogens are. Pathogens. Okay. I heard bacteria. What else? And of course the virus. Viruses. I don't know about virus now. Parasites. And there's another one. Fungi. Funguses called fungi. Fungi is a potential pathogens are things that make you sick or ill or infect you. You know, bacteria would be things like tuberculosis is TB is a bacteria, strep throat is a bacteria, staph is a bacteria, like a skin infection. Those are bacterias. Pneumonia can be bacterial. Viruses are, of course, the flus and the current virus, and measles, mumps, you know, all the common flus, colds. Your cold is a virus. There's lots and lots of viruses. Pneumonia can be viral. Parasites are Giardia, say, or worms. You have worms, or it's like some kind of animal in you, some sort, or it's like a microbe that, you know, Giardia is a microbe. There's dysentery is caused by an amoeba. That's a parasite. Those kind of things are parasites. Funguses, that would be ringworm or athlete's foot. Those are funguses. Actual funguses can infect you. Okay, people get funguses in their ear or their eyes. Amazingly. Okay, those are pathogens. It cause disease, causing organisms. All right, active immunity. A couple more terms. Active immunity and passive immunity. Active immunity is when your body is actively making, you've been infected and your body is actively making an antibody to a specific antigen. You got infected with a virus and your body's reacted by making an antibody to that particular virus. That's active immunity. Passive immunity is when somehow you've been given, let's do this. This is either due to immunization. When you're immunized, it stimulates active immunity. It causes you to make antibodies to that pathogen, or it's by infection. The actual organism. Passive immunity would be when somehow you are given antibodies. That could be an immunoglobulin shot. That's you've been passively given them. They only last for 3 weeks or so and they're gone. You're a fetus getting antibodies for mom, or you're an infant breastfeeding and getting antibodies in the breast milk, that kind of thing. Okay, those are passive. Passive immunity doesn't last long. That's its disadvantage. But the advantage is you don't have to be infected with the organism or whatever it is that could potentially infect you. So you don't have to have the disease to get the immunity. You're just being given the antibodies. Now you have the immunity. There is immunizations like that. There's a tetanus shot. Tetanus is a bacteria, by the way. There's a tetanus shot that there's two types of tetanus shot. There's a tetanus shot where you're given the antigen, the tetanus antigen, and your body responds with active immunity. And there's a tetanus shot where they give you the antibodies to tetanus in the shot, the actual antibodies. So one of the advantages to that is it's immediately functional. When you're given the antibodies, if they're immediately in bloodstream and they're immediately functional. So the advantages of passive immunity say you don't have to have the disease. It's immediately functional. The disadvantage is it doesn't last long. It only lasts 3 weeks or so because those antibodies are cleared. The advantage of active immunity is that it lasts sometimes a lifetime. You get the memory cells that can make those antibodies for a lifetime. So long-term immunity. The disadvantage, of course, is you have to be exposed to the pathogen either by immunization or by having the disease. Now, the immunizations, they give you the dead pathogen. They don't give you the live one, so it doesn't cause the disease. It's dead. But sometimes, somebody could have a reaction to that. It's not the actual pathogen they have a reaction to, it's the preservative in the shot. That's, 1 in 10 million or something. It's very super, super rare. I haven't known anybody that's ever been hurt by an immunization in my whole lifetime. Okay. All right, let's go to some more terms with immunity. Let's see here. Humoral immunity. And cell-mediated immunity. Humoral immunity that's referring to antibody-generated immunity. You have plasma B cells cranking out antibodies. That is termed humoral immunity. Cell-mediated is when you have say a cytotoxic T cell that is destroying a pathogen. It punctures its membrane, puts a hole in it, and water goes into the cell and explodes, if it's a bacteria. That's cell, it's mediated by a cell. It's mediated by the T cell, okay. Cytotoxic T cell. So this is through antibody. It's distant. You make the antibody, it flows around in the blood, goes to the site of an infection, reacts with the pathogen. Cell-mediated, the cell actually has to be physically stuck to the pathogen to puncture it, to destroy it. Cell-mediated. All right. The primary response and the secondary response. Let's do this first. Let's do the clonal selection theory. The clonal selection theory is what they believe is the way that you develop immunity to pathogens during your lifetime. You make a clone. You've been selected to make a clone of antibody-making cells, certain plasma B cells to a certain pathogen. Clonal selection. So here's how that works. Say you were infected with a certain virus. You have been born with, remember there's plasma B cells and there's memory B cells. What kind of cells are those? Those are all lymphocytes, right? They're all lymphocytes. That's the two types of lymphocytes. The memory B cells are lymphocytes that are they're like dormant plasma B cells. They're not actually making the antibody right at the moment, but they remember how to make a certain specific antibody. And they can be in circulation for almost a lifetime, if not a lifetime. So they circulate around in blood for many, many, many years. And if you get infected with that pathogen, then they're activated and they become a plasma B cell and they start cranking out an antibody for that specific pathogen. It's a memory cell. Those are the kind of cells that are stimulated by an immunization. Causes your body to make a memory B cell to, say, polio, which is a virus, or tetanus, which is a bacteria, or measles, which is a virus, like that. Plasma B cells then are the lymphocytes that are actively making a specific antibody to a specific pathogen. Okay, so here you are, you're infected with this flu virus, and you haven't had this flu virus before. It's a different one. And so your body now has to design that plasma B cell that it hasn't had before. So now we are born with memory cells that are similar to the types of antigens we could encounter in our entire lifetime. So there's always a memory cell that we're born with that's similar to the type of plasma B cell that we need to fight off that particular pathogen. So now we're not born with enough of them for each type of pathogen to confer, to provide complete immunity. We're not born with enough of them of each of the types. So when we're exposed to that new flu virus, that virus we haven't had before, our body has to kind of alter the genetics a little bit and then manufacture a plasma B cell, can make an antibody to that particular flu. So that takes a little bit of time. That takes some time. And, you know, hypothetically, and during that time period, that virus could proliferate and kill you. So the response is slow. Now you have other cells in the meantime that are attacking. You have neutrophils and eosinophils and macrophages, and so you have other cells that are battling that pathogen while your body is making the antibody plasma B cell. So it's not as though you're completely vulnerable. So you have an army there battling. But the antibody response really is that's the most powerful immunity you have. So once you get that antibody response, then you typically get well, get over that infection. It's just that we have to get to that point. It takes 3 weeks or so to get those antibodies, 2 to 3 weeks. So, you know, once you develop that immunity of those plasma B cells, and then you develop the memory B cells to that particular flu, then you won't get that flu again in the future. If at that same flu you come in contact with it, you won't get it again. And that's the whole thing with the flu shot. They give you a flu shot. It gives you 4 flu viruses that are common for that particular year. Here's the thing with the flu, it changes each year, double crosses you by changing each year. It's different. So your immune system has to start over again. But if you ever come in contact with that same flu again, you won't get it. Somebody else might get it because they never had it before, but you won't get it. You see, that's the good thing. So when you get a flu shot, they give you 4 viruses. They're dead. They call it attenuated. They're dead. They can't give you the flu. People, if they get a flu shot and then they get the flu, they blame it on the flu shot. It's not the flu shot. It's the coincidence that you got the flu in, you know, like I say, it took 3 weeks for that antibody plasma B cell population to develop. So here they got the shot, and then a week later they got the flu from somebody, and they didn't have the antibodies yet, so they developed the flu, and they blamed it on the shot. It's not it. The other thing is, the other thing is that your body's response to a pathogen is, a fever and body aches and pains, all the things you get, kind of some of the symptoms you get with real flu. That is your actual immune response. So you get a flu shot, and sometimes you get a little bit of a fever, and you get body aches, and so you feel like you got the flu, but you don't. What that is your body's immune response. You're feeling your body's immune response. Now, I know I had the flu. I would get the flu every single year as a teacher, from the people in the front row of the class, every single year, once or twice a year. And about 12 years ago, whenever that was, when there was a swine flu, that terrible swine flu, H1N1, I got that. And oh man, I was so sick. I was so sick. 104.5 fever. That's medical emergency. It kills people. And it lasted for a month at least. It was terrible. And then after that, I decided, oh man, you just need to get the flu shot from now on. And so I did. I have not. Every year I get the flu shot for 12 years now, and I have not again had the flu. I got it every single year up to the flu shot, and now never. So it works. I don't get colds either. It's conferred, there's a cross immunity thing, and it has worked in me. So I don't even get the colds thing. I don't get viruses anymore. It's changed my life. That one year I was talking about, I actually got the flu 3 times. I got the seasonal flu, I got the H1N1, and I was on a vacation where, like Yosemite, where there was a lot of different people from other countries. I got some foreign flu. And so I was sick, 3 months out of that year. Now I'm never sick. Amazing. So the flu shot works. And we all know now with this current virus that we're just all waiting for that vaccine. We know how important a vaccine is all of a sudden. A lot of people realize the importance of a vaccine. The vaccines that we have, sometimes parents choose not to give the vaccines to their children. There isn't any more smallpox, so there isn't any more polio, but there is. But the reason it's safe for those people who don't get the shot, it's relatively safe because everybody else has had the shot. And so everybody else doesn't get sick to pass it on to them. So it's kind of a social responsibility for everybody to get the immunizations, and then nobody will get the disease. It works that way. Okay, there are, by the way, if there's 1 in the 3 million or whatever it is, 10 million that has a reaction to the vaccination, it's because they had a reaction to the preservative that the vaccination was in. And you can actually now request to get single shot immunizations, which mean that they have created that one shot for one-time use, and it's not in a preservative. So there isn't the preservative there to react to. Anyway, let's go to the primary response and the secondary response. So finish off the clonal selection theory. So when you're exposed to that antigen, that pathogen, your body reacts. It alters a memory cell that you were born with into a cell that can make an antibody to that particular pathogen. And then those cells proliferate. They divide and they make a bunch of copies, identical copies of themselves. There's the clone, a big giant clone of these plasma B cells. And they're all cranking out antibodies, and this happens in about 3 weeks, and 2 to 3 weeks, and you get over the disease. You make enough antibodies to get over the disease. But that didn't happen the first time through. the first time, I mean, it did happen the first time, but it took the 3 weeks. So here you go, primary response and secondary responses. The primary response is just what I described. The first time you come in contact with a certain pathogen, your body has to take time to change a memory cell into a functional plasma B cell and make enough of them, make a big enough population of them to fight off the disease. That's the primary response. So the first primary response is slow, takes about 3 weeks, and then you have tons of antibodies. The secondary response, now you have created these memory cells from that first infection or that immunization. You've created the memory cells. And now they're with you for lifetime. Sometimes you have to get a booster shot, but sometimes they're with you for life. And whenever you come in contact with that pathogen, your body has memory cells that just quickly become active and divide quickly and crank out millions of antibodies, and you don't get the disease. It fights, you actually were infected with a little tiny bit of the disease, but it didn't last long. It lasted a few hours. It's gone. Now, that doesn't mean you were sick for a few hours. It means that you had it in your body for a few hours, but then you made enough antibodies to clear it up, and it never proliferated enough to make you sick. So you never got the symptoms of the disease. That's the secondary response. So that is the strategy for all immunizations, to put you through the primary response, to develop those memory B cells that can be functional quickly in your future if you ever come in contact with that disease. Now I mentioned that there was 2 different types of tetanus shots. There's a tetanus shot that puts you through the primary response. That's the common tetanus shot we all get. Get your tetanus, diphtheria, pertussis combination shot. Pertussis is actually whooping cough. That's something a baby could get. Get that if you're a parent, so that your babies don't get whooping cough from you. Anyway, so the tetanus shot, you get the shot, takes 3 weeks. Now you have all your memory cells created. Now you have immunity to tetanus. And when you get an injury, that's the tetanus type injury. You got scratched by an animal or bit by an animal, or you scratched yourself on a dirty fence or something in a farm, got an injury, a puncture wound. Now you potentially could have been exposed to tetanus. And tetanus is this fast killing thing, man. It can kill people relatively quickly, a few days. Now with the tetanus shot though, you've got these memory cells that are responding within hours, getting rid of tetanus before it has a chance to proliferate, kill you. But now what if you chose not to get your tetanus shot? I'm not going to get my tetanus shot. I don't take the chance. And now you get this injury on your farm, or you get bit by a dog, or you get scratched by an animal, something. Now what do? You don't have your memory B cells yet. A regular tetanus shot would take 3 weeks and the tetanus will kill you in the meantime. That's not going to work. So what do? Well, here's the other strategy. You make a tetanus shot that's got tetanus antibodies in it. So what the medical profession does is they take tetanus, put it in a horse. Horses are big, they're huge. Give them a tiny little bit of tetanus. Their body responds to it by making tetanus antibodies. They live through it. And then you take their blood and you extract the tetanus antibodies. They're the same as ours. They're a mammal. The tetanus antibodies from the horse. Put it in the serum, in the shot, full of tetanus antibodies, and you give it to that person that had the injury that didn't get their first tetanus shot. And that'll give them immunity. And you give them both shots. So then you give them the other shot too, the regular shot that works in 3 weeks. Give them both shots. And you got the one shot that's got the antibodies that gives them immunity for the time being, and then the other shot that takes over in 3 weeks. And you get a booster every 10 years and you're clear of tetanus. So people, we don't have anybody with tetanus anymore. It's because of that. But tetanus it's not like tetanus is uncommon. If it is common, you go sometime. Go in Marysville here, say go to the Marysville, the old Marysville Cemetery. Go there and read the headstones. And you'll see that many of those, there's all people from the 1800s and so forth buried there. You'll see, oh man, a high percentage of them as being children. They died under 10 years old. They died of infection. They died of bleeding. They died of some kind of disease that we now have an immunization for. They died of it. So, we don't realize until now, I think maybe the world is realizing how important a vaccine is with this COVID virus. That goes to, that's how it functions for all of those diseases. When we don't worry about tetanus because we get shots, we don't worry about the measles or the mumps or polio or smallpox or diphtheria or all those things that killed people 100 years ago. That they had 50 different types of viruses and bacteria running around in the world like the COVID virus all at once with no vaccines, no protection. Imagine that. Not just the one virus, but all kinds of them ready to kill you. So we think about it. Okay. Thanks for listening. That was about 50 minutes, and that's your immunity lecture for Bio 35. Thanks for coming.